So this is your practical schedule. So gram stain, very important. You need to differentiate between gram positive and gram negative before you can proceed on to the next identification step. Microscope, you have to know how to use them properly. Okay, it's a skill to be able to focus. Okay, using a microscope. Okay, so it's a skill that when given a chance, you must try to master the skill of focusing on the microscope. So pore plating, spread plating for the enumeration of the bacteria. Then dilution streaking is this way. Then for bacterial identification, you can check out the different types of colony. These are terms that you can use to characterize your colony. So if you are doing bacteriology work, you need to characterize your colony to see whether it's a round entire margin, whether the margin is lobate, undulate, whether it's flat, raised. You have to look carefully because all these colonies can be different. Okay, colonies from different bacteria can be different. Flat, raised, convex. When you're working with environment bacteria, you'll see that different types of species can really give different colonies on different shapes of colonies on the general media. Okay, so all this will be helpful for you to characterize your colony. So spore staining, important if you need to differentiate between spore forming and non-spore forming. Helpful, especially more likely for gram-positive bacteria. But it's good to practice more on it also. MFT. Okay, so this one uses a gridded filter. So this is called a gridded filter. The filter comes with grids. Okay, it's to help you with your counting. Sometimes when after filtration, you'll find that it is difficult to pick up your filter because the vacuum is still on. There's still vacuum here, so your filter is a bit more difficult to pick up your filter. You have to off the vacuum or unplug this so that the membrane filter is easier to pick up your membrane filter. Then acid fast, very important for Mycobacterium tuberculosis and Mycobacterium leprae. Okay, so usually we report them as acid fast bacilli or non-acid fast bacilli. Most probable number for identification counting of water samples, and these are the tests that you all have gone through. We've gone through this. Do you have any questions about all these biochemical tests?
No.
Then bacterial identification, API 20E kit. and enterotube. Enterotube is like this. So there's a needle here, and you have to poke it all the way through. Then also there's a code for it. Then from the code ID value, you can see the presumptive organism or identification of the organism. Okay, so this is the different. You can see from here it's media based, is agar. Okay, so these are the types of tests that is available. This is for API. Okay, so anaerobic culture, uh, as you have learned, some bacteria grow better without oxygen. Some bacteria grow better with increased CO2. So increased carbon dioxide, we can use the candlelight jar culture. So these are for Capnit bacteria. For example, your Campylobacter and also your Neisseria. Then although we are not working with anaerobic bacteria, Streptococcus, Streptococcus pneumonia, Streptococcus species, many of them are microaerophilic, and they will actually grow better in a anaerobic condition. So we can have an anaerobic chamber or anaerobic jar. For practical, we use anaerobic jar. So we will place the plates in first, and in the anaerobic jar there's this palladium pellets. So these are catalysts for the reaction to convert the hydrogen and oxygen to water. Okay, so if you're going to a new lab, or if you're going to a lab that is not run properly, you have to make sure that the catalysts are still inside, okay, and it's still active. You can check it by having an anaerobic indicator strip, which is essentially just methylene blue. So it will be colorless. The methylene blue will be colorless if there's no oxygen. So if you cannot buy this strip, you can also prepare your own methylene blue solution and put it here. Okay, just to check it out. So if you're using anaerobic jar, once you place the plates here, then you clamp it. You have to put in the gas pack. Okay, activate the gas pack, put it inside, then clamp it up. Okay, tighten it up, and then there will be a reaction in the gas pack. The reaction is the gas pack contains sodium borohydrate, bicarbonate, citric acid, and cobalt chloride. So this one will release hydrogen and CO2 gas. Okay, so the hydrogen will react with the oxygen, existing oxygen here, will react with the existing oxygen and to form water, but only with the palladium catalyst. Okay, so that's why when we are activating the gas pack, make sure you don't do this near the flame because hydrogen is dangerous. Okay, so you don't do this near the flame. You activate it and quickly put it in the anaerobic jar. That's why sometimes after incubation here, you'll see water vapor at the side. Okay, it's okay. It's just that it's just a reaction between hydrogen and oxygen forming water. Okay, so the anaerobic indicator strip is helpful to see whether your jar is anaerobic. Okay, so if your anaerobic gas strip shows oxygen, that means it might mean there's a leakage somewhere. But if you look at this process, you realize that on many occasions the bacteria here is exposed to oxygen. That's why if you're working with an anaerobic jar like this, most of the time you will only get uh, aero-tolerant anaerobes, you will get microaerophiles, you will get spore-forming anaerobes. Okay, it's very rare for you to get true obligate anaerobic bacteria, for example, Bacteroides or Fusobacterium, because this bacteria die quickly when exposed to air. So once the sample is here, at these stages, they would have probably died. Okay, that's why, as I mentioned earlier, anaerobic microbiology is a very specialized field, need specialized. That's how you use the anaerobic jar and also the candlelight jar culture. Okay, these are some of the questions that you should be able to answer. That you are okay. From your observation, so if you're looking at you're doing spread plating and pore plating, what do you think are the differences? Same
Method.
sample, yeah?
Method.
Yeah, but from your observation, the method is different. So, uh, what other things are different?
The colony of the bacteria form.
Yeah, what's the difference? What's the difference?
Spread plating, the colony only form at the top of the agar, but pore plating, we can see the colony inside the agar.
Okay, very good. Do you think there'll be a difference in the count?
I think pore plating will get a bigger amount because the quantity we take from the sample is 1 ml. But
No, yeah, but when I said, do you think there'll be a difference? Means after enumerating to CFU per mil, do you think there'll be a difference between pore plating and spread plating?
Yes.
Um, what difference?
Uh,
Which one is higher?
Power plating because they have more access to the nutrients, right?
More access to nutrients.
Because if you do spread plating, they're just on top,
Yeah.
whereas the power plating, they can get the nutrients from the bottom of the plate as well. Does that make sense?
Oh, which means they can get the nutrients from everywhere lah, surrounding them lah.
Yeah. Is
Okay,
that
but
right?
I think I
yeah?
think pore plating will get better, much colonies because if we make spread plating, we only use inoculum, right? So we only take a small amount.
Yeah, but as I mentioned, we compare only after we obtain the CFU per mil.
I think it should be the same.
Let me try. I think for plating will give less colonies because uh, we use molten agar, so the heat may kill the cells, so there'll be less colony. That's my that's what I know. Yeah.
Yeah, very so theoretically there shouldn't be any difference because if you're counting the same sample. But of course, if you are as what Iril is it? As what one of your colleague, your classmate said, molten agar can kill some of the more sensitive, heat sensitive bacteria. Okay. But from the past experience where we compare these measurements, we don't really see any difference. But it is very different the way they grow when they are inside the agar and the one growing on the agar. So if you are really planning on continuing to work with them, like if you're planning to isolate them and then continue purifying them, identifying them, it is much easier if you use spread plating because it is very difficult to actually break the agar to try to pick them up and all that. Okay, so these are all considerations that you should take into account. Okay, are there microbes in the mineral water and drinking water sample? So what we will usually ask the students to filter when they do MFT, they will filter some mineral water bottles, and some of them will filter the water cooler, the water sample from the water cooler. So do you think there are microbes there? They'll usually sample about 500 mils of water.
Surely there is, right? But so little.
Pardon?
Surely there is, but very little amount.
Yeah, there will be bacteria, yes. But so the good thing is having bacteria doesn't mean that all are pathogenic. Okay, so you must remember that. Okay, not all bacteria will immediately cause disease and all that. And also, whatever you drink, it goes to your stomach, acidic stomach. Okay, you also have an immune system, so it's not not so not it's not the 100% case of getting diarrhea just because there's one or two bacteria. Of course, you are also exposed earlier today to the infectious dose. Okay, there is always an infectious dose, which means the bacteria has to have a certain amount before it can start causing problems. Okay, next, discuss the advantages and disadvantages of the four enumeration techniques. Okay, so I've already mentioned spread, pour, MPN, and MFT. Okay, tell me, anyone, advantages and disadvantages among the four techniques.
MFT is for large volume. That what I remember.
Yeah, large volume, but you must remember what large volume of what? Of cleaner
Clean
waters.
water, yeah.
It's better if you use the word cleaner, because you do not want to have problems with pores clogging up. I mean, your filter clogging up. Okay, so so if you talk about you can filter lots of water, then you must link it to detection limit. Okay, always link your when you're comparing methods for enumeration, you must link it to detection limits because that is the more important point. Okay, what else? The advantages and disadvantages of these four methods. So these are all four of them are enumerating culturable bacteria. Okay, all four of them requires culture. One of them is probability based, MPN. The other three are what you can consider as more true estimates. Okay. What else? So three of them are plate solid media based. One is broth, right? MPN is broth, right? So MPN broth is better to resuscitate stressed bacteria. Broth is also better for particulates or food-based samples, right? Because it's very hard to spread, plate anything with so like molten agar stress, heat stress, thermal stress. Spread plating will have a bit of physical stress as you're spread plating. Then you can talk about logistics and practical constraints. When you talk about MPN, a lot of cubes you have to handle. Okay, a lot more work. MFT, the problem is always with clogging. Okay, so a lot of times when you use river water, on paper it's supposed to do well, but it doesn't work really. It doesn't really work well in Malaysia because our waters are so much more full of a lot more particulates, a lot more suspended solids. Okay, you must remember most of the standards operating procedures and all this are developed in temperate countries where their waters are cleaner, more pristine. Okay, so we have to take all this into consideration. Anything else? So y'all think about this, okay? Then we are supposed to write a report on the identification steps. Okay, this one. So dichotomous key. So if you are given a bacterial isolate, okay, you most probably you'll be given the bacterial isolate on a McConkey agar. So immediately you know whether it's a lactose fermenter or a non-fermenter, right? If it's a lactose fermenter, it will be pinkish reddish. If it's a non-lactose fermenter, it will be colorless. Okay. Then from the size of the colony, you can guess whether they are actually Enterobacteriaceae, or not, because McConkey has bile salts. So those that can grow well are usually those who have adapted to bile salts. They can, which means from the intestinal tract. So usually gram-positive cocci are all very small because they don't grow as well. But Enterobacteriaceae, they will grow better. Okay. But yeah, then first step you do is what? As a bacteriologist, you will always do gram staining first. Okay, gram stain. So if you get a gram positive, then that's a different set of tests. If you get gram negative, then you know, oh, it's a gram negative lactose fermenter. So what do I need to know next? Next thing, probably I will do an oxidase test, right? Oxidase test. If it's oxidase positive, immediately I know it's not Enterobacteriaceae. I know it's most probably either Vibrissaceae or something else. Okay. Yeah. So or Pseudomonasaceae. Okay. So there are certain tests that you can do. So if it's a lactose fermenter, it's pinkish and it's oxidase negative, then you can do the IMVIC test, indole methylate, Voges-Prosker, and citrate utilization test. That will tell you a lot. If it's positive, positive, negative, negative, then most probably it is an E. coli. If it is negative, negative, positive, positive, then it's more likely Enterobacter. Okay. So these are all dichotomous key steps, right? Step by step by step. Okay, it takes time. Yeah, but for identification, practical purpose, it is still valid. But in the clinical settings, in private labs, you won't have the luxury of time. Okay, most of the tests will be done one shot. They'll use the blunderbuss approach. They'll have the diagnostic system ready already. Okay. Okay, you need to know these principles behind each identification. Any questions about this part?
Nope.
Okay, so this is a hemocytometer. This is a glass slide like with drains. The drains at the drain here, these are drainage. These are to remove the excess sample from the grid. Okay, you're supposed to place a sample onto the hemocytometer, then cover it gently with a cover slip. Okay, so the excess water will drain here. Then there is a standard fixed volume of water in the grid, volume of sample in the grid. Then from there you can count the bacteria in the grid. Since you know the volume of the grid, you can estimate the the bacterial count in your uh, liquid sample. Okay, so how does this differ from your spread plating, pore plating, MFT, and the rest? How does this microscopic count on the hemocytometer differ from spread plating, pore plating, MPN, and MFT?
They're estimates.
Anyone?
Are they? Are they?
Yeah?
Are they like estimates? Like the other ones, you count them one by one. Right.
They're not growing. The
Uh,
others, you need them to grow. Right. But this
Yeah.
one, no, you're counting them directly. Okay, so that's the tip. So that means some of the bacteria that will not grow on your plate, will not grow on your media, will still be counted. So if you're using hemocytometer direct counting, the counts will definitely be higher than your culture count. Because a lot of bacteria actually do not grow on artificial media. Okay, please remember this very important point, because in bacterial enumeration, the first step to differentiate bacterial enumeration is whether you are counting colony, which means culturable counts, or direct counts, direct total counts versus uh, culturable colony forming units. Okay, so that is a very important uh, differentiating factor between them. So you have different grids depending on the type of hemocytometer that you buy. Okay, I personally prefer this improved Neubauer, because this is on a the visibility here is much better than the other grids. Okay, so